Welcome, everyone. On behalf of the American Committee and the Weizmann Institute, thank you for tuning in and participating. For those of you who may not know me, I'm Dave Donison, the CEO of the American Committee for the Weizmann Institute of Science. We are very excited to host all of you today. Very few of us would have imagined just a short time ago that our headlines and televisions would be filled with news such as the COVID-19 global tracker. Could wearable technology be the next tool to stop the spread? Bending the curve, preventing the overwhelming of our healthcare systems, or the one that piqued my interest this morning, our best defense against COVID-19, science. As an organization that has supported the Weizmann Institute for more than 75 years and committed to the credo, science for the benefit of humanity, I am both startled by the furious pace of the epidemic and its global impact, as well as cognizant of the legacy of our three-quarter-of-a-century mission to nurture, build, and sustain a world-class leader in the exact areas of science that are now most needed. Currently, in response to the pandemic, the Weizmann Institute is pioneering ways of massively scaling viral testing on campus, using AI and computer science to help predict viral outbreaks, using computational and structural biology to identify or design therapeutics, working on vaccinations on multiple fronts, and pioneering efforts at leading distance learning for the children that are not allowed to attend school. If the strategy of the Institute since its founding is providing the scientists the freedom, infrastructure, and creative outlets to tackle the deepest and most pressing scientific questions, the current crisis has unleashed a deluge of inspired, promising, and purposeful scientific collaboration in response to this heightened sense of urgency. In record time, the Institute is doing what it does best: collaborating, sharing, adapting, and doggedly pursuing the knowledge that will eventually vanquish this pathogen. And at the same time, the ferocity of this virus is being felt. Many of us know colleagues, friends, and family members that are personally impacted by the virus. This is becoming very personal, very fast. And that is why it is so incumbent for us to win this scientific battle. And to describe in some detail the broad strokes of this, we are joined by two scientists from the Weizmann Institute this afternoon, who can detail for us the Institute's international and national leadership in scientific excellence and research. Our main speaker will be Professor Benny Geiger, Professor of Immunology, and VP of the Institute, Professor Roei Ozeri, who is helping to lead and navigate the quickly evolving situation. Professor Ozeri was born in Israel and earned a Bachelor's of Science in Physics from the Hebrew University. He received his master's and his PhD degree in physics from the Weizmann Institute. He conducted postdoctoral research at the National Institute of Standards of Technology in Boulder, Colorado, in the group of David Weizmann, a physics Nobel laureate, and he joined the Weizmann Institute faculty in 2007. Roei.
Thank you very much. In the last 2 or 3, even in the first week of this pandemic, we saw dozens of initiatives coming from scientists on campus, taking their research and steering it to tackle the coronavirus problem. We have a large activity of life sciences on campus, so people have many expertise who are relevant to this pandemic situation. Most of them from the life sciences departments, many of them coming from computer science departments, even from physics, from chemistry. People are trying to take their knowledge and direct it towards trying to find one solution or another. And we see initiatives going in directions of trying to find vaccines very quickly, trying to do drug discovery very quickly, improving on our ability to expedite and increase the volumes of coronavirus tests that we can do. And some of the initiatives, and Benny would tell you about the initiative that he and Eran Segal are leading, uses computer science tools and questionnaires in order to try and detect and focus on outbreaks of the coronavirus without actually having to go through biological tests. We as management are trying to support these initiatives to the best of our ability. We understand that here we're at the forefront of the struggle. I think this is the money time of science. I think that's very clear. And I think it really varies. But if some people or some governments were not so happy to support science, now it's very clear that science is, everyone's eyes are turning to science to try and find the answer, and we understand the responsibility, and we understand this is our call. It's a challenge because at a time when we need to reduce our personnel to the 30% level, we still need to put our foot on the gas pedal and try and drive ahead as fast as we can. So again, we're trying to support these initiatives, both logistically in terms of releasing the personnel that is needed, also coming up with the funds that are needed to carry out this research at the necessary speed. And this is also the reason for us opening Weizmann Coronavirus Response Fund, through which we're trying to support the research which is conducted on campus. And I think this is extremely important. So with that, I'd like to move and introduce this evening's speaker, Benny Geiger. It's a true pleasure to introduce Benny. Benny is one of the pillars of life sciences at the Weizmann Institute. Benny joined the Weizmann Institute as faculty in 1977. He's a cell biologist and studies the interaction of cells with their microenvironment. Benny published more than 400 scientific papers, which is a lot of papers. And I think Benny served probably on almost any administrative role at the Weizmann Institute you could imagine. He was the head of the Department of Molecular Cell Biology, the dean of the Faculty of Biology, and currently we recruited him again to serve as the head of the Department of Immunology. Up until recently, Benny was also the chair, the head of the Israeli Science Foundation, and tonight Benny will tell you about his new initiative to battle the coronavirus. Please, Benny.
Thank you very much, Roy. Indeed, I mean, we are in a very, very, I think, special and unexpected and trying period, which is very unusual to all of us. To many of us, it is quite frightening. And I have to admit that for many of the scientists in a whole variety of fields, not only in biology or biomedicine, but also in computer sciences and in chemistry, in physics and so on, this period is offering fantastic, amazing challenges. Because in our encounter with this virus, we have a lot of unsolved questions and problems, and the knowledge about the virus and its behavior and its future is extremely unclear at this stage. To some extent, we are looking now at a situation that started to be investigated seriously during the last few weeks. And we are collecting a little bit information from previous SARS epidemic and so on. But altogether, we face a lot of new problems, new issues. And to some extent, interestingly, this is for us a challenge that we feel comfortable with. I mean, this is our sort of comfort zone to encounter problems that are unresolved. That's what we are doing in research in general, and try to quickly get to understand them, know what this virus can do, and so on and so forth. So in order to get you acquainted into the system and provide you some hints, really the tip of an iceberg of what's being done at the Weizmann in Israel in general. So the idea, what I'd like to tell you a little bit about is what we are doing here at Weizmann as a community and as individuals about it, and to some extent to what level this epidemic has brought many of the researchers within Israel to network together and work together towards making this epidemic as bearable as possible and end up in a favorable situation for Israel and for the world worldwide. And as you'll see, I'll try to start from individual laboratories and go through some of the major projects that we are leading here and put some attention into this particular study that Eran Segal and we are pushing forward. The virus itself, in general, a family of the corona is quite well familiar to us for a number of years, mainly through the SARS epidemic period. But here we are looking at a virus that is not very different from the one of SARS. It's about 80% similarity, almost identical to what was called the SARS CoV2, which was a virus responsible for the SARS epidemic. This one is closer to a virus that was identified in bats, in pangolin, and it is known in a certain detail. We know what type of proteins it consists, we know what it recognizes in the body, but how altogether it acquires its particular properties that make it different from other viruses of the family and make this epidemic much more difficult to control and really develop into a pandemic affecting essentially all countries is something which we are starting to understand and to investigate as we go and enter into that field. So the virus is, of course, tiny. The size is about 1/10 of a typical intestinal bacterium. And it has a variety of components. The spikes are important not only because it gives us a nice name, coronavirus, looking at the shape of a crown, but these are the structures that enable the virus to stick to the cells in the upper respiratory system and actually cause a disease. I mean, each of one of those can bind to specific molecules that are located in our upper respiratory system, and through them, the virus enters into the cell and modifies its activity. We still don't know what part of the disease is attributable to damage that the virus itself is causing, what part is being contributed by the response of the body in attempts to fight against the virus, which can also have an effect. Nevertheless, this virus is really, in many of its features, is new to us, very infectious. It's not causing, in many cases, in most cases, a very difficult disease, but it is extremely infectious, and those people who are infected, some of them have other issues, age, and some background diseases that make this virus potentially lethal to them. Now, when this virus is entering into our respiratory system, it makes contact with the wide variety of cells within and on the way to the lungs. And then there is a progressive process that the body in most cases can handle, but in a significant number of cases it cannot, and then we get a disease that can be fatal. So this is the basic information. By the way, all the slides that I show you here and the references that are relevant, if you're interested, we can send you, because I think in some of them there is some new information that is really changing from one day to the next. So what's actually happening here? When the SARS CoV2, which is the cause of the COVID-19 disease, when it started to develop in China and started to show features that are really epidemic or pandemic features, Israel was aware of the risk that it will penetrate into Israel, either directly from China or through other countries. Eventually, most of it came through Europe. And the challenges here were multiple, which had to be dealt with by the Ministry of Health as the body responsible for taking care of such situations. One issue was a question of diagnosis. How do we know that the person has the SARS CoV2 and not other disease like flu and so on? And there were tests for SARS, which could be in a way adapted to deal with COV2. In SARS it was COV1. But still at the stage that we understood that the disease may penetrate, these diagnostic tools were very slow, very cumbersome, quite expensive. The process was, there was equipment that was badly needed in order to do it. And I have to say that still in Israel and in most places, the diagnostic part is an issue. The policy of the Ministry of Health was to try to identify people who are sick and isolate them. But in diseases that spread and increase rapidly, it is not less important to find out what is the percentage of carriers of the virus in the population. And this could be done only if one could really do many tens of thousands of tests per day, which is still away from us. And here we at the Weizmann Institute are trying to deal with this issue, as you'll see in a minute. The other issue is that the infectivity is very high. The disease is spreading rapidly. This virus is not as frightening as many other viruses that we know from the literature and from the medical experience, because there are some that are really much more lethal, but the speed at which it expands is something which is unprecedented. And therefore, the question of how can we block the spread in the population was an issue. Obviously, the lockdown strategy, quarantining people, is one way to go. But as we'll talk a little bit later, I mean, this has its limitations. Also, the question of the prominence in Israel in terms of knowing how many people are infected at any given moment and are moving because they don't know that they have the virus. And we also know that, that the symptoms can be very highly variable. And being so variable, it is difficult to figure out who needs to be treated at a very early stage. We already see situations of people dying here and in many other places who were not really categorized as strong and clear candidates for developing COVID-19. So all of that situation and the fact that we have kind of a poorly defined enemy makes it very difficult for the ministry to really have a strong policy of how to move, and the decisions are being actually developing and changed from one day to the other, which is also happening in many other countries. Of course, we are in a way lucky that the disease started here a little bit later than in some places, and we are learning the lessons that were taught by those experiences. But as I mentioned before, most of the scientists, the situation of uncertainty is a comfort zone, because we have here a possibility to really perhaps change things, especially because we know how to ask questions and to address them, and in an unorthodox manner. And that was the reason that when we realized that we are facing a serious problem here in Israel, there was initiative in many universities. I think the initiative was very strong here at the Weizmann Institute. And I sort of participated in a meeting of researchers, a group of researchers, about 10 of them, mostly from the Weizmann, a few from other universities, which we convened with the Ministry of Health, with the leadership of the Ministry of Health on March 11. And we actually, frankly, asked them, and we told them, look, we know that this is something which is very challenging for you. We need to know what kind of help do you need that we can give you. And the answer was very simple, and I think the same answer was given also to Alon Chen, who was talking to the government about this issue. And the need was for collaboration. There are certain aspects and approaches for understanding what can happen, which would be very difficult for people who are not professional researchers in this field to really take. There was a need for infrastructure loan or support of infrastructure, because definitely the current infrastructure in hospitals is insufficient to cope with the challenges. The people who are trained to do these kinds of analysis and tests are people usually who practice standard processes in life sciences, modern life sciences, and there was a great need for volunteers who will operate testing system that will enable to upscale the discovery of the virus. We need a focused research on the short and long-term properties of this epidemic. And there was a great need to help with predictions so that we can be prepared not only for the current situation, but for what can happen in the near future. And definitely we have to quickly try to develop new therapies because epidemics or pandemics of this nature is usually not short-term, one-season diseases. They are there to continue, and if we want to really deal with this virus, we need to have a long-term solution like vaccination and so on and so forth, which need to be developed. And the anticipation is that the next target time will be the beginning or the late fall and the beginning of the winter 2020, which is not a lot of time for developing therapies. And for all that activity, it was clear that we need to have between the scientists massive networking between scientists from different fields, different institutions. I have to tell you that what I personally witnessed during those days was something which was really, for me, unprecedented. I mean, there were many lines of discussions by WhatsApp and by other means between scientists from different places, including scientists from outside Israel, that were dealing with specific questions. And I think part of it was manifested in a very, very dramatic fashion in something that I don't expect you to read, but just it's a table that shows the recruitment, self-recruitment of laboratories within the Weizmann Institute to different aspects of coronavirus research. Though I think within about a week, the institute in which there was one single laboratory that was dealing with the SARS at the time, so it was sort of half relevant to the current corona epidemic. There was over 50 projects that were already initiated by March 27, which is really, really, truly dramatic. And just to cut this long list short, they dealt with diagnosis, with handling the virus, with drug discovery and different types of therapies. Many of them were dealing with big data technologies, to which people coming from the artificial intelligence field and the computer sciences and mathematics were participating, physicists, and people in structural biology. And many of these studies are now already initiated within the Institute, and the idea was not to wait until there are funds for that in the usual process, but really move forward very, very quickly. Now, one of them, which is a major project, was, I think, the decision to open within the Weizmann Institute, was sort of in a country that is short of testing virus stations and working relatively slowly, to open in a very efficient manner a facility for testing for the virus. What you see here in the picture is Robert Flo, who is running the INCPM, the GINCPM, the Nancy and Steve Grant Center for National Center for Personal Medicine. Part of the equipment was moved into a specific part of the building. People were trained to do the tests. We are now in the process of getting the formal approval from the regulator, from the ministry. You can see here in the picture Robert and the part of the team at the INCPM just getting the instructions of how to deal with safety issues since the virus actually reaches the place when the virus is still active. And I think this was very dramatic, very inspiring. The numbers of tests that will be done is not clear yet, but we are trying to think big and to try to build it to fairly large numbers. We're trying to think critically on how to do it so that we may be able to improve it. And then the test will be carried out by students from the Weizmann and postdocs and people from the staff of the Weizmann Institute. So this is a major, major undertaking that we have initiated here, and probably in the next few days, this activity will start. Now, another approach which dealt with some of the shortcomings of the current technologies is being approached by a new team of 2 researchers and their groups. It's called kind of the ACE team, which is Amit Corona Elinav, Ido Amit and Neral Elinav, who had an interesting idea. I mean, they were looking at the current procedure, and the current procedure, which is, taking a sample of the virus, some cells from the patient. The problem is that it's a labor-intensive, manually carried out process, which is unscalable. I mean, you cannot really run it at a high throughput. There is a global reagents shortage. It's expensive. It's unsafe to both collection and to the lab staff. It is molecularly a complex process that includes biochemical purification of RNA from the virus. It has a reduced sensitivity. It's expensive in time and so on. And the idea or the objective of Eran and Ido was to move everything to automation, to develop a new approach, a new technique that can bypass the slow processes, the slow steps in the current activity, to use robotics that will enable robots to collect the materials and not to be manually dealt with by people. And the new technique will be based really on this kind of automation and should be user-friendly. Now, there's something like that to develop it usually takes years. And the question was whether this can be done in a couple of weeks. And actually, that is a mission. For that, they needed some equipment. So right now, they have in a special big laboratory that was furnished and built for them in one of the buildings at the Weizmann, they needed equipment. So this is a robot that is called Benny because it was taken from our laboratory. Maya was taken from Maya Schuldiner, Amos from Amos Tanai. So they have a collection of equipment that was taken from running laboratories. The principle was to do everything without needing to purify RNA, which is a slow process and time demanding and expensive. And by this kind of robotic flow of processes from Benny to Shalev to Maya, and from Amos to Maya, and then do additional analysis, put them into the computer, process the data, and have a rapid diagnosis, and basically move this diagnosis. This diagnosis reaches immediately the Ministry of Health, which can give the instructions to the physicians what to do, in principle, getting protective measures. So this is really an amazing, very, very quick process that within days was established. You can see here the the lab, the different robots that are located in the lab. This is, I am sentimentally attached to this one, which was taken from here. And you can see here the people properly dressed and working on this project. So this is quite amazing. One of the first things that they did was suggesting a process whereby you can kill the virus before it enters the lab. So this is something which is very important in order to keep the safety of the people working in the building and the safety of the staff that is working here. And this is already a success. And now I think the process starts to move into the practical parts. The person in disguise here is Ran Elinav, and you can see here the volunteers that work in this particular project. So this is really a very heavy undertaking, and I think this is going to, if it works properly, it will be a revolutionary process. Now another story which was in which I was, or not is, I am involved, is an idea that was sort of raised in a discussion in the morning of Friday, March 20th, by Eran Segal and myself. The idea was that it might be extremely important to try to use big data technologies, artificial intelligence processes in order to be able to forecast, to predict which regions in the country are more prone to show outbreak of the disease. And the idea was to distribute over the web, distribute as quickly as we can questionnaires that people will report about their health. The idea was to do it in a way that people will be attractive to people to respond to, and it will be informative enough in order to be able to draw conclusions out of it. So the idea was to predict the future local increase in disease, collecting information about current symptoms, then develop internet-based questionnaire, use powerful artificial intelligence to generate the available predictions. And it was clear that this had to be coordinated with the Ministry of Health. So it should be, the whole system should work together. We are joined by Yuval Dorf of the Hebrew University, who had very strong suggestions of how to complete this. And you can see the questionnaire over here. It asks questions about the age, male or female, the rough address, not the exact address, because the people remain anonymous. And with our efforts so far, you can see some more questions about the health situation. There are about 400,000 people have responded to it, which is nice. It's insufficient to be fully effective, but it is nice, and it already gave strong predictions that convinced the Ministry of Health that this is an important investment, and we are now moving into a much broader framework that will bring in, we hope, something in the neighborhood of 1.5 million to 2 million people to provide daily report about their health. And just to give you a demonstration, what you can see over here is kind of a map of prominence of people who complain about symptoms that are relevant to the coronavirus infection. You can see it in the map of the entire country. Here you can see the neighborhoods around Haifa, and if you go down here, you can see even small regions within the city. This is kind of downtown Haifa, in which there are higher situations of disease presence. And just to illustrate to you, one of the things that you may, if you follow the news here, you may hear about, if we look on Tel Aviv area, there are two regions that are that are much more active in terms of complaints about symptoms. And the prediction, which was done on March 23rd, was verified by actually reports from clinics in these regions a day later. So it gave us a day in advance in order to get ready, to get prepared for the situation. We believe that with larger numbers, we'll be able to have predictions that will be even more accurate. Now, we are working together with the Ministry of Health to quickly build the entire system into their computer system so that they can respond to the predictions made by Iran's group and by us. Another approach that out of really the 50 that I mentioned before, another approach is approach that was sort of initiated by Amos Tanai, who is working on and using very, very heavy computational approaches to biomedicine for a while. Most of his activity until recently was sort of directed towards question of genes and genomic characterization of disease. And here he sort of felt that it would be very important to identify and understand areas that are the areas that are responsible for spreading, for viral spreading. In a situation of a lockdown that we are facing. So these places are the supermarkets, usually the food chains, because no matter, I mean, people even if they are locked in their homes, eventually they have to eat. And these are places where people who may be carriers of the virus can distribute the virus, and definitely specific peoples within the shop, usually the cashiers, can be the critical vectors of passing the virus. They have the highest chance to be infected, and then they have the highest chance to infect other people that come close to them. You cannot really deal with the cashiers from a distance of 2 or 3 meters. So they have mapped all the supermarkets in Israel. The idea was to go to 5,000 buyers and store employees, do viral testing for them, follow them later, and figuring out what are the pathway through which what are the spreading avenues of the disease, if you wish, and try to find ways of blocking them, which can be extremely useful and extremely important. And another issue, which sometimes you have to think about the disease not only for its biomedical aspects and health aspects, but also from other side effects, which is the risk that a lockdown for a long period of time can really destroy the economy. And it's a lot of discussion of how do you balance the needs of keeping the epidemic low and not letting it expand, and at the same time keep a sustainable kind of sustainable economy, where originally physicist and systems biology expert Uri Alon, who moved into biology quite a number of years ago, tried to ask, I think, an interesting question. He said, well, do we really, if we have a lockdown, does the lockdown have to be a hermetic lockdown? Or can we get kind of effective reduction in viral spread by having an kind of intermittent lockdown? And he tried to run a variety of models of disease spread and said, well, what happens if we are, instead of having a continuous lockdown, which obviously will bring the disease to a reduction, what happens if we let people, for example, work for 2 days and then or 4 days, 3 days or 4 days, and then be locked for a certain period of time? And they worked in a weekly kind of intervals models for 5 days lockdown, 2 days work. And it turned out that under those conditions, the disease will diminish over time of a few months. If, for example, instead of 2 and 5, they will go to 3 and 4. The disease will continue to rise and increase in volume. So this distance, this particular formula of 2 and 5 worked well. And then he said, well, it doesn't have to be 2 and 5. It can be also 4 days and 10 days, or 6 days and 15 days, which you can see over here that will lead also to a decline of the disease. This is something which is a formula that is discussed in different, with people in the government, and there's a lot of interest in finding out kind of sustainable solution in the long term. And I think the last example that I'd like to give you and close with this is an initiative that is really looks to me like a unique networking effort, global networking effort that was pioneered by Eran Segal and a few colleagues around the globe, Feng Zhang and many others, which is encapsulated, if you wish, in the few sentences that I highlighted here, that information is the most potent weapon we have to combat a pandemic at both the individual and global level. And there is a kind of a white paper that was issued now, creating a group that will be the core of a network that will be able to deal with both modeling and both by understanding the biology behind the process quite well, that could learn from experiences in other countries that can benefit the world at large. So these are just a few examples, and I think many others are just starting now. And it's clearly what keeps many of us busy. It's what's going to happen towards the winter of 2020. And I'd like to close here and thank you. And if you have questions, I'll be happy to answer. And in order not to finish with a picture of a virus by electron microscopy, I'll show you how the GI and CPM looks when viruses are not around. So thank you very much, and I'll be happy to answer questions if you have any.
Excellent. Thank you so much, Benny. The first question that I thought was highly relevant was you talked about the ACE protocol, the high throughput screening that has been put together on campus at Weizmann. And the question was this protocol being shared with other countries or other institutions like the United States?
The idea in general is known as being shared. The protocol doesn't exist yet, but the protocol is not something that you write it on a paper and you have a protocol. This protocol has to work, and I think they are working, around the clock in order to get a protocol. It's obvious that once the protocol works in a way that can be used by others, it would make very little sense to try to start in parallel to develop the same protocol in different places. So I think we are now already witnessing very, very significant progress. The system is calibrated. The system very shortly will be tested on certain volumes of patients about what can be the throughput of the approach. We are talking about days. And I think when this is working, it will be shared immediately. And I think there are contacts all the time with other universities here and with many, many colleagues around the globe who have input into this activity. So it will be shared, I believe, with everybody.
The next question we had was just an inquiry. Is there a chance that people who have had the pneumonia vaccine are faring better than those that haven't, meaning the COVID-19 has less symptoms?
I don't think we know that by now. It's what we do know is that usually there are antibodies. Well, let me clarify, give a little bit of detail how it actually works. I mentioned to you in the first couple of slides that the spikes on the virus are attaching to cells in our body, in the respiratory tract. When there are antibodies, most of the antibodies actually bind to the spike and block the spike. That's like, if you're like putting a glove on my hand so that the hand cannot directly touch another surface. Now this, we know that antibodies appear in the SARS epidemic, and also in the COV2. They appear within about 10 days after people start to show symptoms. We don't always know when the people were actually infected, but when they show symptoms. Initially, there are two types of antibodies. There are antibodies that come initially, then different type of antibodies come a little bit later. We don't know yet how protective these antibodies are and what can make them more protective. So, for example, there is this particular issue is being investigated here by one of the scientists at the Weizmann, Weizmann, who is a structural biologist, who is knowing how antibodies can interact and block parts of viruses. That's something that he was sort of working on and dealing with in the past. And now he is directing to, in a way, redesign antibodies so that they can fit the ones that are known from to work in SARS, will fit to the virus by fitting them like really a glove to a hand onto the new virus. So I think part of it will be done by kind of synthetic antibody production. Some of it will have to be done through some kind of immunization. There's a lot of activity in that direction in Israel at large. I mean, there is a vaccine to the chicken form of corona, which done in a laboratory in Galilee. There is the Institute, the Israel Institute of Biological Research in a nearby city, Nes Ziona, not far from the Weizmann, 3 minutes drive from Weizmann. They are working on a vaccine. There is a lot of activity, and I think one of the longest lists that was in the initial projects that are starting now to take place at Weizmann have to deal in one way or the other with the vaccination and with things that can be done like towards passive vaccination, namely prepare antibodies that will be used to block a disease and an existing disease. I think that's also something which will be attempted.
Great, then Benny, the next question is, there's certain parts of the world, whether it's Israel or Spain or Italy, that we're hearing very troubling stories. And I'm in New York, so certainly we're hearing a lot of news about the spike there. But then there are other places like Africa that we're not hearing a ton of information. Is that because that there's a lack of testing? Is it just a matter of time because it gets there? Is there a difference in temperatures? Do we have any sense why there's a difference here?
Well, first, I think the information that we have is limited. It's true that I think from the little we know is that there are certain regions that are not really visited by foreigners so much. So they are in a way isolated because they are always isolated. I mean, there's not much communication, and there I think there is no sign of disease. Usually the places of disease in Africa start in places that are being visited by people from the outside, and the information coming from there, definitely they don't have the diagnostic tools to do much about it. And in most cases, it's okay because, I mean, many people recover from the disease. It could also be that there are some environmental factors. I mean, this is a cold favoring disease, and Africa is still great parts of it. The temperatures are higher, which makes it much prone. But beyond that, my guess is as good as everybody else's.
The last question I'll ask is, there's a new Abbott test from Abbott Labs that's supposed to be coming out this week. Have you heard about that, Benny? It's supposed to be very fast.
We heard about that. We didn't see yet the way in which it works. I think the problem with some of the kits that provide you the information, a rapid diagnostic tool like the kind that is being used for pregnancy tests, etc. You get a blue line. It's very difficult to figure out what is their threshold. So the question is, which by the way is true also for the sophisticated methods, the routine methods that are using molecular biology to do the analysis. The critical thing is to identify also very low levels of contamination. Because, I mean, today it can be a low level, in 2 days it can be a serious disease. The sensitivity is definitely still a big challenge, and I don't know at all how sensitive the Abbott approach is.
Excellent. I do want to ask Roe, if you have a couple, give you a minute or so to make any observations or closing comments that you'd like.
Yes, I can say a few words about Nir London's project. Not that I'm an expert, I'm a quantum physicist, but I attended several of his web seminars. So Nir is an expert on computationally designing and then experimentally testing molecules, chemicals that can affect the way proteins act, pathogens often. And very quickly after the outbreak in China, a collaborator of Nir in Oxford was able to reconstruct the structure of the protease, which are the scissors that come packed with the virus. Nir's group was able to very quickly come up with 30 different leads to chemicals that can affect the action of the protease. None of these 30 chemicals can actually prohibit the protease from going into action, but they're a starting point. And therefore, they decided together with Sloan Kettering and a couple other institutions to initiate a crowdsourced type project where they gave these leads as an open source. They also gave their computational resources as open sources. And they asked medicinal chemists from all over the world try and come up with combinations of these 30 different leads to come up with possible drugs that would prohibit the protease from going into action and therefore would disable the coronavirus. After about a week of collecting ideas, they came up, so they received more than 1,000 different suggestions for drugs. They're now trying to prioritize these suggestions using their computational tools. And after prioritizing, they're sending in the priority list these drugs to be synthesized by an industry that agreed to do that at their cost expense. And after these are synthesized, they'll check them one by one in labs, trying to discover a drug that would work. Trying to take such a massively parallel approach into drug discovery is not the traditional route. In the example that Nir gives and his collaborators is it's like, when you play golf, you try to drive the ball into the hole by hitting it multiple times, getting closer every time. The approach they're taking is like taking 1,000 golfers trying to hit the ball and hit it in a first attempt right into the hole. It's a moonshot. It's not an easy attempt, and if you want to go full scale, it'll require a few millions of dollars, $10 million, and we're helping Nir and his collaborators get the funding which is necessary for this massively parallel drug discovery effort. So this is a short description of what Nir is doing. I wanted to close and thank not just all of you for joining us tonight on this Zoom web conference, but all of our philanthropic partners for your support throughout the years, and especially during these stressful times. Just saw a live example in Benny's talk how the GINCPM was established through the generous support of the Grant family and many other families is now at the forefront of our struggle against the coronavirus. So your support means a lot to us. It always has, and it continues to be. So thank you, and wishes of best health.
I want to thank everyone for your participation, your great questions. Again, I apologize for not being able to get to all of them in the interest of time. We'll have more opportunities to do so in the future. I want to thank Benny and Roe and all of the scientists and students at the Weizmann Institute for their heroic efforts in fighting this global pandemic. I can just say that after the last hour together, I find myself feeling more hopeful, a bit more optimistic about our future and the ability of the best scientific minds collaborating on the vital tools of science to defeat this deadly enemy. On the behalf of the American Committee and our global network of donors and supporters, we're proud to be on your team, and we will do whatever we can to help you succeed. To that end, I just want to reiterate what Roe mentioned, which was last week we announced a global fundraising drive to raise $25 million for research related to the novel coronavirus, which has basically begun to be funded. I'm proud to announce that we've already received several $1 million of funding since beginning this important initiative. And I encourage all of you who are interested to reach out to your local Aquis professional, or you can write me personally. You now have my email if you're interested in getting involved in this global effort. Again, I just want to wish everyone, I hope that you're safe and you're healthy, and I look forward to the next time when we can all actually physically be together again.